So now we're going to talk about gametogenesis. Gametogenesis is the formation of specialized sex cells or gametes. An egg cell would be formed as a large sessile gamete, and that means sessile does not move. And sperm cells are the formation of smaller motile gametes. So motile means to move. So what is spermatogenesis? So it's the production of sperm in the male gonad, and this happens in the testes. So it's the development from unspecialized cells called spermatogonia. Primary spermatocytes form the spermatogonia. During the first meiotic cell division, the primary spermatocyte produces 2 secondary spermatocytes. During the second meiotic division, the 2 secondary spermatocytes produce 4 spermatids. The spermatids develop into mature sperm cells. Some animals only produce sperm during specific mating seasons. In other animals, such as humans, sperm are produced all year long. So this diagram is showing you spermatogonia. Replication, which means now we have DNA replication. Meiosis I will occur, and the sister chromatids are going to separate, so the homologous pairs will separate. And then during meiosis II, we're going to have the sperm cells, which are 23 for humans, chromosomes in each sperm. Now we're going to talk about oogenesis, the production of eggs or ova in the female gonad, which is the ovary. Mature eggs develop into oogonia, which enlarge to form the primary oocyte. The primary oocyte undergoes the first meiotic division, producing 1 secondary oocyte and 1 smaller polar body. So remember, before in lecture 2, we discussed what happens during cell division meiosis II for males and females. This is where we have a difference. So the secondary oocyte undergoes the second meiotic division, producing 1 otid and 1 polar body. All polar bodies will disintegrate. The otid grows into a mature ovum or egg cell. The ovum is a non-motile cell, meaning not moving, that contains a small quantity of stored food in. And in this diagram, you're able to see that the primary oocyte replication through interphase, DNA replication, goes through meiosis I with 46 sister chromatids in those homologous pairs, divides, and of course goes through meiosis II, and we have a single egg cell with 3 polar bodies. And please remember that those polar bodies are going to disintegrate. Now, how does fertilization occur? Well, first, there has to be union of a sperm cell to the egg cell. So it occurs when the sperm cell penetrates the membrane of an ova or the egg cell, and the haploid nucleus of the sperm unites with the haploid nucleus of the ovum. Ovum is going to be our egg cell, forming a diploid zygote nucleus. Now, internal fertilization, eggs are going to be fertilized within the human body. Specialized organs are going to be needed in order to deliver the sperm cell from the male body into the female body. Fewer eggs are going to be formed, but many sperm. Mostly this will be found among animals that reproduce on land. There are some aquatic organisms, including the shark and lobster, that also perform internal fertilization. External fertilization. So eggs are fertilized outside of the female body. Each parent releases their gametes directly into the water, and the sperm will use the water to swim to the egg. This will take place in animals that breed in water. So some examples would be salmon, coral, and frogs. So what do you think some of the hazards are of external fertilization? I'll give you a moment to think about what some of those hazards are. So one could be that the sperm and egg never actually meet, the eggs could be eaten, or a temperature change could actually cause cell death. Then we have parthenogenesis. This is the development of an unfertilized egg cell into an adult animal. This is a type of sexual reproduction. However, it's no sex, please. So what's going to happen usually occurs in insects, which will be invertebrates such as bees, wasps, amphibs, and ants. But it can occur in sharks and snakes. So in bees, the queen bee mates one time. Then she produces either unfertilized or fertilized eggs. The unfertilized eggs become male drones, while the fertilized eggs become female workers, or some will become queens. So what are the advantages? Well, they can reproduce even if there's no males present. The disadvantages, however, are less diversity. So that means there's going to be less pups able to fight infection or able to deal with challenges that arise from their environment. So why is meiosis a source of genetic variation? Well, independent assortment. A pair of chromosomes can be arranged in two ways when they pair up at the start of meiosis. The analogy, it's like shuffling a deck of cards where each card is a chromosome. Homologous chromosomes would be the chromosomes with the same color and number. The cards are shuffled and dealt to two players. The result, 8 million different possibilities. So how would more pairs of chromosomes affect the number of possible arrangements? I'll give you a moment to think about that. Well, hopefully everybody said increase. So the more chromosomes, the more possible arrangements. Then we have crossing over, which is when homologous chromosomes form tetrads. The strands of the chromosomes can twist around each other and may exchange segments. Then we have genetic recombination, so recombine to combine or recombine differently. So when a sperm fertilizes an egg, the chromosomes are recombined. The diploid chromosome number has been restored in the zygote. However, there may be new combinations due to the variations that were passed down from mother and father. So there's about 64 trillion different possibilities when a sperm and egg cell unite. So now you're going to receive a test yourself, and hopefully you understand the major differences between mitosis and meiosis.